Hello everyone, I'm Clémentine Tetaert, and so today I'm going to present you my master's thesis that is currently under review and which is entitled Functional Adaptation of Ankle Bones in Proboscideans. So we know that in terrestrial quadrupeds, body mass, among other factors, plays an important role in the morphology and microanatomy of the appendicular skeleton. And today I will talk about some heavy specimens that are called graviportal. So the concept of graviportality was introduced at the beginning of the 20th century and still has the controversial definition. However, some criteria have been agreed upon. So a graviportal taxon must weigh at least several hundred kilograms, have columnar and robust limbs. So columnar limbs are limbs that are aligned vertically under the body, like you can see right here, as opposed to the flexed limb of cursoidal taxa. They also have relative reduced distal bones as well as large feet with cushion pads. So today I will concentrate on the model group of the true graviportal taxa, which are proboscideans. And so today the diversity of proboscideans is relatively small, with only 3 species, 1 in Asia and 2 in Africa. But the fossil record is much richer, with around 10 families, 45 genera, and 185 species and subspecies described. And this group is characterized by a great variation in size and body mass, with the first representative being relatively small. You have here the example of Meuritherium, compared to the largest size estimation that has been made for proboscidean, which is Paleoxenon namadicus, which is pretty tall and pretty heavy. And there are also several cases of insular dwarfism within this clade. A recent study by Camille Bader, looking at the morphological adaptation of long bones to high body mass in proboscidean, highlighted the presence of 2 main morphotypes, one represented by Elephantidae and Deinotheridae, which have thin diaphysis and narrow epiphysis, and the other one represented by Mammuthidae and Gomphotheridae, which have robust diaphysis and wide epiphysis. So we can see here that within proboscideans, there are 2 different weight-bearing strategies. And so we can ask ourselves what happens on the autopod side if the same morphological variation and same morphotype will be present or not. And so this was the context in which my study was carried out. So the aim of my study was to explore the morphological variation of the astragalus and calcaneus in proboscidean through the prism of size, body mass, posture, and phylogeny. So the astragalus and calcaneus form the first row of the tarsus, which connect the zygopod, tibia, fibula, to the rest of the autopod. And they are highly involved in the ankle joint, as the astragalus form the center of rotation and the calcaneus the lever arm of this articulation. My sample consisted of 149 3D specimens, including 71 astragalus and 78 calcaneus, which represent A family and 34 terminal taxa. The method that I used in my study was the 3D geometric morphometrics analysis, using 3 different types of landmarks that, when combined, can quantify the overall morphology of specimens to then compare them. And I also performed several multivariate statistics that I will develop during my presentation for greater clarity. So first, I'm going to talk about the result of the allometry analysis. So allometry is the covariation of size and shape, so it allows us to see the influence of size on the shape of an object. For the astragalus, we have here a significant evolutionary allometry, which accounts for about 6% of the global morphological variation of these bones. And we can see that the increase in size, which corresponds also to the increase in body mass, has an effect on the articular facets in red and on the ligament essential zone in blue. We obtain the same result for the calcaneus. And thus, in heavy specimens, the astragalus and calcaneus have wider ligament essential zone in blue and articular facets in red, which allow for better transfer of axial load as well as better interbone support. We can also see that heavy specimens have a shorter astragalus neck, which is this structure right here. and thicker tuber calcaneae, which is this part. And those structures are area of fragility on those bones. So it consists on a reduction of these areas of fragility. And you can also see that there is more medial impact of the increase in size on these bones. Then I estimated the phylogenetic signal linked to the K variable to see the influence of the phylogeny, the implication of the phylogeny in morphological variation. And so we can see here that we have a significant phylogenetic signal, but the value of the K molt well below 1 suggests morphological convergence among taxa. Finally, I carried out a PCA. So in this figure, you can see the first two species, which account for about 24% of the global variation. And the heat maps help us visualize the structures that vary the most along this axis. And so we can see here that the structures that vary the most are mostly articular facets, and the ligament insertion zone, which is the medial tubercle right here, and those structures are wider on the specimen distributed in the most negative values of both axes. For the calcaneus, I'll concentrate on the first species, and we can see that the structure that vary the most is the tuber calcaneus, this structure right here, which is the muscular insertion zone on which, among other, the gastrocnemius and soleus muscle, which form the Achilles tendon responsible for the plantar flexion of the ankle inserts. So the plantar flexion is the extension of the ankle. And so the specimen distributed in the most negative value, which are Denituridae, Amebulontidae, and Gomphotheridae family, are a more elongated and thinner calcaneus than the other specimen. And so when we look at the distribution of the specimen on this morphospace, we can see a separation between two groups. The first one represented by Elephantidae, Mammuthidae, and inborn Stegodontidae, and the other group formed by the Deniotheridae, Amebulontidae, Gomphotheridae, and the Coirolophonditidae families in yellow. And so I wanted to test whether the two groups that I saw were significantly different. So I ran a MANOVA on the first two axes and the groups that I identified and came out significantly different. And so I also wanted to see if those groups were applicable to the astragalus morphospace. So I tested it and it came out significantly different too. But here we were only testing on a small part of the variance, so I checked the difference taking into account 100% of the variance with Procrust ANOVA, and the result are significant for both bones. So I now consider them to be two different morphotypes that have similar composition in terms of size and body mass. And since the astragalus and calcaneus are highly involved in the ankle joint, these morphological differences may have an impact on the locomotion of the specimen, particularly during the plantar and dorsiflexion, so extension and flexion of the ankle. In fact, the size of the tuber calcaneus, which is the lever arm of the joint, has an effect on what we call the effective mechanical advantage. So a longer lever arm leads to a higher EMA, and thus a better amplitude capacity of movement than a shorter lever arm. And so to compensate for the lower EMA of the joint in the M2 morphotypes, we can see the recruitment of more muscle, ligaments, and wider articular facets. So we have the M1 morphotype, which seems to be associated with better ankle mobility for plantar and dorsiflexion, and the M2 morphotypes with better joint stability to compensate for the loss of mobility. And finally, I compared the result between the long bones and the arthropod for both 2D. We identified two main morphotypes, so we could think that they will be represented by the same group of taxa, which is not the case here. There are several combinations between the long bones and tarsal bones within a single taxon. So we have different patterns that do not match the phylogeny, which could reflect different waist-bearing strategies, different locomotion, a different way to have the same locomotion, or a mixture of everything. And this could also indicate different stress integration between long bones and tarsal bones, with long bones morphology more influenced by body mass, and tarsal bones morphology more influenced by environmental factors, such as substrate or type of environments, which was also a hypothesis for all other taxa. So I will skip my conclusion because I don't really have the time, but it's the same thing that I just said. So basically it's okay. And thank you for your attention.